Welcome to the Biopharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com.
Phase 3 clinical trials and the road to FDA approval. SEPA fourth, executive summary. Drug development is a long and fragile process. A promising molecule can survive years of preclinical and early human studies, only to fail at the very last stage. Phase 3 clinical trials are the true crucible. They determine whether a therapy is not only biologically active, but also effective, safe, and feasible for real-world use. This article explores how Phase 2 signals are translated into Phase 3 confirmation, the strategic choice between placebo and active comparators, the spectrum of endpoints that regulators demand, and finally, the structure of a new drug application, NDA, before the US FDA. One from Phase 2 to Phase 3, a structural leap. Phase 2 and Phase 3 trials are often conflated, but their functions diverge sharply. Phase 2 is exploratory. 100 to 300 patients, surrogate endpoints, multiple dosing arms. Its goal is to ask, does this drug seem to work, and at what dose? Phase 3 is confirmatory. 1,000 to 10,000 plus patients, randomized and blinded, with hard endpoints such as survival, hospitalization, or major adverse cardiovascular events. The question becomes, does this drug change outcomes at scale safely enough to alter medical practice? A successful Phase 2 trial may excite investors, but only Phase 3 data can build the evidentiary foundation for approval. 2. Designing a Phase 3 protocol, from signal to legitimacy. The protocol of a Phase 3 study is not written in isolation. It is the direct translation of Phase 2 findings into a decisive test. Dose selection, chosen based on PK/PD and exposure response from Phase 2. Usually narrowed to one regimen, multiple doses dilute statistical power. Population, broader than in Phase 2, with stratification by age, sex, comorbidities, or biomarkers. This ensures generalizability. Comparator, the most strategic choice. Placebo is valid when no effective therapy exists, maximizing statistical clarity. Active comparator, gold standard, is required when withholding standard of care would be unethical. Here, trials test superiority or non-inferiority. Endpoints. Regulators demand hard outcomes whenever possible. Surrogate endpoints are accepted only if well-validated: HbA1c, LDL-C, blood pressure. The estimate framework, ICH E91, defines exactly what treatment effect is being measured and how to handle events such as rescue therapy or dropout. Statistics, alpha control, multiplicity adjustments, interim analyses, and predefined margins for non-inferiority. Phase 3 must withstand intense statistical scrutiny. Safety. Detection of rare but serious events requires scale. Independent DSMBs monitor ongoing data with authority to stop the trial for futility or harm. 3. Placebo or gold standard, the strategic divide. Choosing whether to compare against placebo or the current gold standard determines not only feasibility, but also regulatory perception and market positioning. Placebo, allowed when no standard exists or when withholding therapy poses no undue risk. Statistically efficient and often faster. Example, molgramostim in autoimmune PHAP used placebo as no approved therapy was available. Gold standard, active comparator, mandatory when effective therapy exists, oncology, cardiovascular, infectious disease. Demonstrates either superiority or non-inferiority plus added value, better safety, convenience, or cost. Example, the CagriSema redefined trials in obesity must compare against semaglutide or tirzepatide, as placebo alone would be clinically irrelevant. The choice reflects not only ethics but also strategy. A head-to-head trial that proves superiority can reposition an entire therapeutic class. Before, what must be proven? Beyond efficacy alone. Regulators such as the FDA do not approve drugs on efficacy alone. They require net clinical benefit relative to existing options. This can be demonstrated in different ways. Superior efficacy, longer survival, fewer hospitalizations, better disease control. Equivalent efficacy with improved safety, e.g., apixaban showed similar efficacy to warfarin but reduced major bleeding. Equivalent efficacy with better convenience, e.g., once weekly or oral dosing instead of injections. Unmet need. Rare or orphan diseases may gain approval on surrogate endpoints with post-marketing commitments. Thus, approval hinges on more than statistical significance. It depends on whether the therapy adds meaningful value to patients and the healthcare system. 5. The NDA, Gate to Market Authorization. The FDA requires a comprehensive new drug application, NDA, to convert clinical data into marketing authorization. The NDA is structured in ICH-CTD modules. Clinical, Module 5, integrated summaries of safety, ISS, and efficacy, ISE, drawing from Phase 1-3 data. Nonclinical, Module 4, toxicology, pharmacology, animal safety. CMC, Module 3, chemistry, manufacturing, and controls, ensuring reproducible quality. Labeling, Module 1, proposed prescribing information with indication, dosing, contraindications, and safety warnings. Administrative, regional, Module 1, exclusivity, pediatric plans, REMS if required. Approval is granted only if substantial evidence of efficacy, usually 2 well-controlled Phase 3 trials. Acceptable safety profile relative to benefit. Manufacturing consistency demonstrated through inspections and validation. Labeling accuracy so physicians and patients understand how to use the drug. Without these, the FDA issues a complete response letter, CRL, a regulatory rejection. 6. Structural risks and symbolic weight, cost and fragility. Phase 3 programs can exceed $500 million. A negative result can erase entire companies. Operational bottlenecks. Recruitment, site management, and data integrity at scale are critical. Symbolism. A successful Phase 3 not only validates a molecule but also transforms a company into a legitimate pharmaceutical actor. Phase 3 is therefore not just a scientific trial, but a test of institutional resilience. 7. Corporate and financial impact of Phase 3 outcomes. Phase 3 trials are not only scientific crucibles, they are also financial detonators. The results of a single study can transform the fate of entire companies and alter the balance of industries. Positive readouts, value creation. A successful Phase 3 often multiplies market capitalization overnight. Biotechs with limited pipelines become acquisition targets for major pharmaceutical companies seeking growth. Example, Immunomedics rose dramatically after its Phase 3 ASCENT trial of sacituzumab govitecan, Trodelvy, in triple-negative breast cancer, leading to its $21 billion acquisition by Gilead in 2020. Negative readouts, value destruction. Conversely, a failed Phase 3 can erase years of investment within hours of market opening. Companies heavily reliant on a single asset may collapse entirely. Example, multiple Alzheimer's programs, e.g. Biogen's aducanumab, prior to its controversial accelerated approval, saw billions wiped from valuation after negative efficacy results. Symbolic dimension. For investors and institutions, Phase 3 outcomes function as binary signals, legitimacy versus collapse. A win validates not just a molecule, but the company's scientific platform and governance. A loss undermines credibility, erodes investor trust, and can stigmatize entire therapeutic classes. Thus, Phase 3 is both a scientific and economic threshold. It is the point where biology, regulation, and capital markets converge, deciding whether a therapy becomes a societal asset or a financial cautionary tale. Conclusion: Phase 3 trials are the threshold of legitimacy in medicine. They demand rigorous design, ethical clarity, and statistical precision, while carrying the highest operational and financial risks in the drug development cycle. The FDA's NDA process ensures that only therapies with proven efficacy, acceptable safety, and reliable manufacturing reach patients. For the public, it is important to understand that the pills or injections prescribed today are the survivors of this crucible. Behind each lies not only laboratory science, but years of trials, regulatory negotiation, and billions in investment. Annex 2 Case Study, rofecoxib, Vioxx. 1. Approval indication, pain and inflammation in arthritis and osteoarthritis. Class, selective COX-2 inhibitor, anti-inflammatory. Phase 3 evidence, large pivotal studies, vigor class, demonstrated strong analgesic efficacy and significantly fewer gastrointestinal ulcers and bleeding compared to traditional NSAIDs. FDA NDA, approved in 1999 as a therapeutic advance, positioned as safer for the stomach and intestines than conventional drugs like naproxen or ibuprofen. 2 post-approval problems. Soon after launch, observational data and extended follow-up trials revealed a marked increase in myocardial infarction and stroke. Why it was missed in Phase 3? The pivotal trials were designed around gastrointestinal safety, not cardiovascular endpoints. The number of cardiovascular events in those trials was too low to demonstrate a clear pattern of risk. Number 3. Regulatory and corporate action. In 2004, the approved trial, adenomatous polyp prevention, showed that long-term use doubled the risk of serious cardiovascular events. Merck voluntarily withdrew Vioxx worldwide in September 2004. The withdrawal affected over 80 million patients, triggered one of the largest litigation waves in pharmaceutical history, and cost Merck more than $25 billion in settlements and lost market value. Number 4. Structural lessons, endpoint bias in Phase 3, focusing on gastrointestinal safety, blinded the program to cardiovascular danger. Scale of exposure matters. Risks invisible in 5,000 trial participants became catastrophic in millions of real-world patients. Regulatory humility. Approval does not mean invulnerability. Robust pharmacovigilance and post-marketing commitments are essential safeguards. Conclusion. Vioxx is a cautionary tale. It passed FDA standards for efficacy and apparent safety, became a blockbuster, and was then removed from the market after real-world data revealed a fatal imbalance in risk-benefit. For the public, it illustrates why regulatory approval is not the end of scrutiny, but the beginning of long-term surveillance.